Hello everyone, welcome to the ITLS Academy. Today we are going to discuss another topic in the series of food preservation, and the topic that we are going to discuss is about the monitoring the effectiveness of the food preservation. Okay, so what we have to do, we have to monitor that when we apply a different food preservation method for the preservation of the food product, so how these methods are effective and what is the outcome that we get by applying a different preservative technique. Okay, so let's start the lecture monitoring the effectiveness of the food preservation. So simply in this lecture, we are going to monitor the effectiveness of the food preservation, and along with this, this preservation of this presentation also cover the comprehensive strategies which monitor the how these food preservation methods are effective, and also included the evolution of the microbial safety, chemical stability, sensory attribute, and the compliance with the regulatory standards, and highlight the traditional monitoring method along with the modern technological innovation which enhance the this preservative technique and also control the spoilage of the food product and ensure that the food are safe and provide the high quality of the food product to the consumer. Introduction. Monitoring the effectiveness of the food preservation is very essential. See, food preservation is simply means that when we apply a food preservation method, then it extended the shelf life of the food product. Extended the shelf life of the food products. Now, how we can extend the shelf life by deterring the microorganisms, and along with it, we also inhibit the enzymatic activity which occur into the food product. Okay, so this effectiveness should be monitored because it maintain the safety of the food product, provide the good quality of the food product to the consumer, and also monitor the shelf life of the food products. Preservation technique, whether it is thermal technique, chemical technique, or biological technique, they can also be failed. Means that suppose that it maybe the thermal preservation technique may be failed, chemical and biological. Why it is failed? Because maybe some human error occur, variability in raw material occur, and due to that malfunctioning. So these are the reason due to which the if this preservation technique should be can be failed. Okay, so regular monitoring help the identification these failure. Okay, if the regular monitoring occur, then these can monitor the failure, prevent the spoilage, and also ensure that the consumer should get a safe food and also they get a highly nutritive product. Okay, these regular monitoring also decreases the waste produced by the failure of these techniques. Next, objective of monitoring. So the main objective behind the monitoring, like we have to verify the microbial control, assessing the chemical stability, and also ensuring the nutrient and sensory quality of the food product. So the main objective behind monitoring the this preservation technique, that we have to verify that the microorganisms which are present into the food product are controlled, and their chemical stability can be accessed, and nutritive and sensory quality are ensure, and also complying with that regulatory standard and minimizing the food waste. Monitoring also support the continuous process. These continuous process improve the this continuous process help in the improvement in the food quality, identification in the trend which may require the process optimization or corrective action so that the failure can be prevented, because we all know that prevention is better than cure. When a spoilage is occurred, then it is very hard to eliminate it until and unless it will affect the human body. So we have to eliminate it before it occur. It reduces the wastage produced by the spoilage and also ensure that the consumer get the safer food product. Now the next is your overview of the food preservation method. So food preservation method divided into the different technique, like your thermal processing, cold preservation, drying, fermentation, chemical preservative, high pressure processing, and irradiation. In thermal processing, we apply a heat, either a mild heat, either a high heat. Okay, when we apply a mild heat and it only targeted the pathogenic microorganism, then that category is known as a pasteurization. Pasteurization is targeted for those for pathogenic microorganism. And in sterilization, large amount of heat is applied with which is targeted on the all type of the microorganisms. Then we apply a blanching process. In blanching, there is a inactivation of the enzyme take place, and when the enzyme is inactivated, the food will not be spoiled. The next process is known as a cold preservation technique, in which we decrease the food temperature either at 0 to 5 degrees Celsius range. Then that process is known as a refrigeration, or either it is minus 18 to minus 22 degrees Celsius, then that process is known as a freezing process. Next category is your drying process. In drying, we uh, reduces the amount of free water present into the food product, and that air drying is divided into again it can be done by air drying, freeze drying. Okay, then fermentation process, we use a biopreservation technique. Then chemical preservatives, like we can use a sorbate and benzoate for increasing the shelf life of the food product. Then high pressure processing or irradiation. Each method are distinct mechanism for inhibiting the microbial growth or enzyme activity, which require to monitor for the effectiveness. Next is your key parameter which we have to monitor. So if we talk about monitoring the preservative technique, so we have a different parameter that we have to monitor. Okay, so we have to monitor a microbial load, we have to monitor a chemical indicator, nutritive retention, sensory attributes, packaging integrity, and environmental condition. These are the factors which we have to monitor when we have to monitor a preservation technique. So in microbial load, we have to monitor the load of the bacteria, yeast, and mold. And in chemical indicator, we have to monitor the pH of the food product, peroxide value, free fatty acid. Okay, then in nutrient retention, we have to monitor the vitamins and antioxidant. Okay, then in sensory attributes, we have to monitor the color of the food product, flavor of the food product, and texture of the food product. Then packaging integrity, in which we have to monitor the seal strength, barrier properties. Okay, in environmental condition, we have to monitor the temperature of the food product, temperature of the environment, humidity and light. So a comprehensive monitoring plan ensures that the overall preservation system function as intended. The next is your microbiological testing basic. So this microbiological testing basic is monitor the load of the microorganism present into the food product. Monitor the load of microorganism present in food products. Okay, so in this category we monitor the microbiological load by a traditional methods like total plate count, most probable number, and selective media or specific microbes. Okay, this type of methods are used to quantify the microorganisms which are present in either in a product or a process, and also provide a direct measurement of the preservation efficiency. Suppose that if we do a plate count method, now in doing a plate count method, we see that the how much microbial load on that particular food product. Okay, if it is beyond the limit, then obviously it means that the preservation methods are not effective. Despite being time consuming, they remain a gold standard for food assessment. But this traditional technique of the microbiological monitoring are what they are time consuming. They required at least 48 hours to get our result. Okay, but it is time consuming, but it is a gold standard because it gives a proper assessment for the food safety. This is about the microbiological testing basic. Then indicator organisms. So there are some indicator organisms like your coliform, E. coli, yeast, mold, and spore forming bacteria. They serve as a marker of process efficiency. Okay, so if we talk about the coliform, E. coli, yeast, and mold, they are very harmful bacteria which are present into the food product. Okay, and also some spore forming bacteria. They serve as a marker of the process efficiency. They are present often as signal contamination or ineffective preservation. But when we regular monitor these indicator microorganisms, they allow a detection of the process deviation before the pathogen can proliferate, because we cannot tolerate a single number of the coliform or E. coli should be present into the either into the food product or either into the your process. Suppose that like just give an example, like suppose that you are working in some hotel industries, and you are just testing a ice bucket. Okay, now in ice bucket, the from the door of that ice bucket you get the E. coli. Okay, now these ice bucket is not safe for the use. Okay, because a single micro or the single unit of this E. coli will not be tolerated. So this is called a indicator organism if it is present in a only single colony forming unit, but it is not acceptable. Pathogenic specific monitoring. So high risk pathogens like your Listeria monocytogenes, Salmonella, and Clostridium botulinum, they all are what they all are come under the category of the critical target, and they have to monitor in a regular basic. Okay, even this listeria can survive under the refrigeration condition also. So if you keep your food and in a refrigerated condition and you think that there is no chance of the growth of the microorganism, then it is not right. You have to monitor it on a regular basis, that Is there is any growth of this Listeria or not? Now the next is your Clostridium botulinum. The spores of the Clostridium botulinum is required for the validation of the heat treatment. Okay, suppose that you do food preservation by applying a heat treatment. Okay, then you test that you test a microorganisms Clostridium botulinum present into the food product. Then you find out that the spores of that Clostridium botulinum is present into that particular food product in which the heat treatment is applied. Now this give a validation test that the proper heat treatment is occur or not. Monitoring the pathogenic specific indicator also ensure that the preservation processes achieve indent safety level. Rapid detection method. So rapid detection method included the PCR based assay, ELISA, ATP bioluminescence, and letter flow test. They give a rapid uh, result for the safety of the for the monitoring of the food preservation. These provide faster, often real-time result as compared to the conventional culture method. Okay, we get a real-time result means that suppose that if you do a testing by rapid detection method on Monday, so you will get that result on the same day, same time. Okay, and it is very faster as compared to that conventional culture method, because it in conventional culture method it takes around 48 hours. They are particularly useful for high throughput environment or when rapid decision making is necessary to maintain the product safety. Trend analysis of microbial data. So trend analysis is involved plotting the microbial counts over the time to detect a pattern or deviation in process performance, like statistic tools control charge, or it is also known as a SPC, that is your statistic process control. These chart can help you for the identification of the gradual changes which occur into the equipment wear, hygiene lapses, or other risks which are very effective to the preservation efficiency. So this is about the trend analysis of the microbial data. The next is your chemical indicators. So chemical indicators like your peroxide value, which give a indication of lipid oxidation, free fatty acid. It is give a indicator for the hydrolytic rancidity or the residual of the chlorine level in water, which access the chemical stability of foods. So monitoring these parameters ensure that the preservatives are effective and the product maintain the safety and sensory quality throughout its shelf life. pH and water activity. So pH and water activity, these are the critical control factors for the microbial growth. Okay, at low pH, it inhibit the growth of the bacteria. Okay, see, if the pH is low, then it inhibit that the growth of the microorganism, inhibit the growth of microorganism. Same with that water activity. If it is low, then it inhibit the growth of the water activity and the vice versa. Okay, so these are the critical parameters for measurement of the microbial growth. Then low pH inhibit bacterial growth while low water activity prevent the spoilage. Monitoring these parameters ensure that preservation method maintain the condition which suppress the microbial proliferation over the time. nutrient and color retention. So when we apply a thermal preservative technique or either the chemical treatment, then both can affect the nutritive value of the food product or even the color of the food product. Okay, so it can degrade the vitamins content, antioxidant, and the pigments. Monitoring the nutrient retention and color ensure that the product meet a nutritional claim and maintain the visual appearance. Okay, so if we monitor the nutrient retention and maintain that visual appeal, so it can ensure that the product should safe. For example, monitoring the vitamin C content in a fruit juices, it ensures that the heat treatment have not excessively reduced the nutritional value of the food product. The next is your packaging integrity test. So these packaging integrity is used to protect the food from the environmental contamination. Suppose that if we keep our food in a open, then there are lots of microorganisms, dirt, dust will contaminate the food product. So we have to pack that food product into a proper packaging so that they avoid the environmental contamination. So there are different tests like your vacuum retention, seal strength, leak detection, oxygen and carbon dioxide composition. They can check the confirm that the packaging material its barrier properties and preventing the microbial ingress and spoilage. Temperature control and logging. So temperature control is very essential for both way. Like if you apply a thermal preservation technique or either you apply the cold preservation technique. In both the category, the temperature control is very important. Data loggers and sensors record temperature over time. So what we have to do, we have to record the temperature of the refrigeration, temperature of the cooking, everything in a data logger. Verify that the storage and transport condition comply with the required ranges. Suppose that you have a raw chicken. Now you have to store that raw chicken at 0 to 5 degrees Celsius and you can store it around 48 hours. Okay. Now the second thing is that if you cook this raw chicken, then you have to cook it until the core temperature of the raw chicken reaches to the 85 degrees Celsius. This is a core. Okay. Now suppose that uh, if you are not maintaining the log, okay, so sometime like it exceeded it, the temperature is extended from 5 degrees Celsius and reaches to its 23 degrees Celsius, and this will reaches till 2 hours. Now at that time this raw chicken is spoiled. So if you are not monitoring them, then you will not get that is there any variation of temperature take place, because the variation in temperature can compromise the microbial safety and even increase the spoilage of the food products. HACCP integration. Monitoring is a fundamental component of the HACCP system, which is very important. Critical control points like pasteurization, freezing, or chemical addition step. Monitoring ensure that each critical control point is maintained within a safe limits, and it help in the prevention of the deviation which could compromise the food safety. Now the next is your verification of thermal process. So we have to verify the thermal processes through a heat penetration studies. Here we verify that the amount of heat which is given to the food is enough for the food preservation or not. And it is verified by the heat penetration, F value calculation, and microbial challenges test. Verification also ensure that the coldest point of the product achieve advocate microbial lethality, while it will avoid the overprocessing which could damage the quality of the food product. Verification of freezing and chilling. Now, after verification of the thermal processing, the second verification is your freezing verification of freezing and chilling. So it can be verified for the cold preservation method, and it can be monitored by involving the uniformity of the temperature, holding items and equipment performance. Ice crystal size and freezing rate affect the texture and microbial survival. Regular verification also ensure that the freezing and chilling are effective throughout the product. Monitoring the drying and dehydration. So when we apply the drying, simply drying is what? Drying means that remove the water content present into the food product. Remove the water content from the food products. When the water content is removed, their moisture content is reduces, their water activity is reduces, and for the growth of the microorganisms water is required. So when the water activity is reduces, it prevent the growth of the microorganisms. Moisture content and water activity are also measured to confirm that the process effectively inhibit the microbial activity, and inadequate drying can also lead to the spoilage or mold growth during the storage of the food product. Monitoring the fermentation and bioprocess. So fermentation rely on beneficial microculture to suppress the pathogenic microorganisms. In fermentation, we use a beneficial microorganisms. Beneficial microorganism means that the microorganisms which are come under the category of the good microorganism, and when we consume that microorganism, they can benefit it can be benefit for us and provide a nutritive value. Okay, so in fermentation we use that beneficial microbial culture, and this beneficial microbial culture suppress the growth of the pathogenic microorganisms. Monitoring involve like the measuring of the pH, lactic acid production, and microbial population dynamic. It all used to confirm the process is effective. Deviation may also indicate that the starter culture failure or contamination. Sensory evolution basic. So sensory attributes like your color, odor, flavor, and texture, they are the sensory attribute of the food product. It can be monitored to detect the spoilage of the food product or checking the quality degradation. Train panels or the instrumental analysis like your colorimeters and texture analyzer is used to provide the objective data for the product evolution. Then accelerated the shelf life testing. Products are subjected to elevated the temperature, humidity, or light, which stimulate the storage condition, and accelerate the test help predict shelf life, identification of the critical failure point, and optimization of the preservation method without waiting for a real time shelf life to the elapse. Packaging and storage condition audit. Regular audit should be performed, and this regular audit verify that the condition of the storage means that the temperature of the storage, hygiene of the storage, humidity of the storage, everything. And here storage condition means that the humidity, oxygen, light exposure, stacking practices, they should be meet according to the specification. Proper monitoring of that storage environment also prevent the quality loss, and it can ensure that the preservation effectiveness. Next, visible sign of spoilage included the gas formation of odors, texture changes, and mold growth. Monitoring for these indicators enables that the early detection of the preservation failures, and it helps the prevention of the distribution which are unsafe or poor quality products. Customer complaint and feedback. Consumer feedback and complaints provide the indirect monitoring of that preservation effectiveness. Report of off flavor, spoilage, or short life highlight potential process weakness and inform the corrective action. Data loggers and lot devices. So modern monitoring uses a lot sensors and data loggers. It can be used to track the different parameters like your temperature, humidity, and other critical parameters in real time. Data is often transmitted to that central system for immediate analysis, altering managers to deviation. Biosensors and smart labels. So smart packaging, which include that the freshness indicators and temperature indicators, they will provide the real-time information which are related to the quality of the food product and the safety of the food product. Biosensors can also use to detect the microbial activity of the food product, chemical spoilage, or gas composition inside the seal packages. Machine learning and predictive model. Machine learning model which is used to analyze the historical preservation data. It is used to predict the spoilage of the food product, microbial growth, or the chemical degradation. Predictive analytics allow the proactive adjustment to the preservation process so that it will prevent the failure of the food products. Remote and cloud based monitor system. So remote monitoring system is used for the collection of the data which we get from the multiple production or storage location. Feeding centralized and dashboard. Alert are sent in case of the deviation, enabling the real time intervention and enhance the control over the preservation process. Integration of monitoring with blockchain. Blockchain can surely record the preservation data. It can ensure that the transparency should not be altered and traceability should be maintained. It also used to provide the verifiable proof about the product which have been stored and handled under the specified preservation condition throughout the supply chain. Regulatory certification requirement. Regulatory authorities like your FDA, FSSAI, Codex, they are required a document which will monitor of the preservation process, comply, demonstrate product safety, build consumer confidence, and ensure that the eligibility for the certification and market access. Documents and record keeping. Accurate record of monitoring result, corrective action, and audits, they are all essential for the safety management. Documentation enables the verification during the inspection and internal audit, and they can support the continuous improvement and initiatives. Corrective action: if monitoring identify any deviation, corrective action like process adjustment, retraining staff or equipment maintenance are implemented. Continuous monitoring support interactive improvement so that it can prevent the reoccurrence of the preservation failures. Cost benefit: effective monitoring. So effective monitoring are used to reduce the risk of the spoilage, recalls, and foodborne illness. It also ensures that the optimal shelf life of the food product, product quality, and consumer safety, minimize the waste and financial losses. Ultimately, it saves the cost and improving the brand reputation. Conclusion: monitoring the effectiveness of the food preservation is very crucial because it maintains the safety of the food product, quality, and compliance. Then, combining the microbial, chemical, sensory technology, monitoring strategy, they all ensure to robust the preservation technique and integrated approach are enables protect proactive management and continuous improvement. Open the door. Now improvement. So this is all about the today's lecture, which is about the monitoring the effectiveness of the food preservation. I hope you will understand, but if you have any query related to this, you are freely to ask me in a comment box, or for a further detail, you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp, and LinkedIn. And subscribe our channel ITLS Academy on YouTube and get our app from a Google Play. You can also ask your query to the helpline number that are already mentioned in your slide. That is 7080833450, and visit our website, that is www.itlsacademy.com. Thank you for watching. Please like, share, and subscribe our channel.